Okay, gracias and hola. So yes, I'm Sara Gianella, and I am actually from the Italian part of Switzerland. So I'm happy to be back in Europe, and I'm also happy to be here and present about my absolute favorite study, the Last Gift program. So I probably don't have to give too much of an introduction to everyone that is in his room, but about HIV and HIV. Really early after acquisition, it spreads throughout the entire body, so that the HIV reservoir is not only in blood, but really in every possible tissues across the body within days to weeks since acquisition. And even when people are on antiretroviral therapy, HIV likes to play hide and seek with the immune system and antiretroviral therapy. And then if people stop, in the vast majority of cases, it comes back. So a lot of the HIV community and research is now going towards trying to target the HIV reservoir to try to find a cure for HIV. But it's very hard to study the HIV reservoir in tissue in living people, right? It's very hard to reach the deep tissue, like you can't really biopsy a beating heart or a brain. And even the autopsy study that have been around since a while, they were not really designed for HIV cure research. First of all, because a lot of the study don't really follow participants close enough, so that, sometimes the last visit before that is like 6 more months or more. So we don't really know if people are on antiretroviral therapy at the time of that. And also autopsies are often not done very rapidly, as a lot of the autopsy studies in the US involving people with HIV have a time to autopsy that is between 12 and 24 hours. And we know that at that time, a lot of the nucleic acid, protein, and cell viability is already not good enough to do a lot of assay. So this is David Smith. He was the chief of our divisions, and he has been my mentor for a very long time. He had this idea in 2017 to start this cohort called The Last Gift, where we follow people that are at the end of life. They are sick with any life-shortening illness like cancer or a neurologic disease or cardiovascular disease. So we are able to follow them very closely during the last months of their life and then perform a rapid autopsy at the time of death. So rapid autopsies are not very easy to do, and I think I underestimated it when we started The Last Gift. They are done mostly in cancer research, and so far there are about 20 rapid autopsy programs in the US, and 19 are for cancer, and The Last Gift started with HIV. And what makes The Last Gift unique compared to cancer is that we collect all of the tissue, because in oncology, most of the time they collect, during the autopsy, they collect the cancer, the surrounding lymph node, the metastasis, so it's much more limited. But our goal is to collect all of the tissues within 6 hours from death, because we know from the literature that this is how, we can optimally preserve the quality of RNA and protein and cell viability. So why terminally ill volunteers living with HIV? Obviously, first of all, because being close to death, we can really follow them very closely, and we can then collect all of the tissues. There are also no expectation of direct clinical benefits. And also a lot of people at the end of life, they are long-term survivors. They have been around for many, many years living with HIV. They lost their loved one and friends. And so really there is this big desire to give back to the HIV research, but really there are not so many studies that are open to sick and old study participants. And so The Last Gift has been really welcomed by the community, and really we saw it as an opportunity to create this new translational research model that we can use to advance HIV cure research. So to give you an idea about the cohort characteristics, these are 30 participants that already passed, and we did rapid research autopsy since 2017. As you can see, most of them were white male, which is not surprising because this reflects the epidemic of HIV in California. They have been on study for a really variety of time, from 1 day to over 3 years. And most of them died of cancer, but we also had other like neurologic complication and end-stage organ diseases. So these are the demographics from the 12 people that are currently on study. And what I wanted to point out here is that we really did a big effort into diversifying the cohort. And right now we have 4 female and 3 Black participants enrolled. And it was not easy because, again, we really had to do a lot of outreach. And in fact, right now, because of the success of the last gift and the importance of diversity in HIV cure research, we are working with 2 sites in Brazil and South Africa to try to replicate the last gift to be able to access more diverse populations. And here is a figure to show you how long most of the participants stayed on last gift. And you can see there are some participants that stay very short time, like basically we identify them, and then a few days later they die and we do the autopsy, and there are participants that stay on study for a very long time. And this highlights how, and many of you are physicians, right? Like how difficult it is to predict how long someone will be around, even when we know that they have a terminal disease. It's very hard to predict the time of death. What makes the last gift so unique compared to other autopsy studies is that we are really able to collect data in a granular way. We usually request all of the clinical data, historical clinical data, and then at the last weeks, we just go regularly, multiple times a week, to the participant's home or hospice, and we collect all of the medication that they take. We know exactly when they took their last antiretroviral therapy, and then we collect regular blood. And here is just an example. This is last gift 1, and you can see that we collected, no, I don't have the pointer, but we were able to collect blood until the very end, the day before his death, we were able to collect blood. And in this particular case, the participant stopped antiretroviral therapy, so we were able to also collect the rebounding virus in the blood. And so you can see how this is a unique opportunity to not only study the tissue, but to compare the tissue with the blood. Here is a picture of our team and during a rapid autopsy. So like we couldn't do the last gift without a dedicated team that is basically on call 24/7, because the vast majority of our autopsy happen in the middle of the night or in the weekends. And so we're basically all on call. And as soon as someone dies, we all run to the morgue and we prepare all of our material and reagents and tubes. And then we just do the autopsy. And usually there are between 9 to 13 people in the room where we extract the organs, we cut them, we put them in all of our various media and flash freezing and formalin so that then the tissue can be preserved and distributed. And the picture on the left, you can see our minute of silence that we do at the beginning of every autopsy, which is really powerful, where we read this little acknowledgement just to acknowledge the gift of every study participant. It's also a really terrific educational opportunity for students to observe such a rapid autopsy. And usually we exit the morgue with over 1,000 tubes of tissues. And then what we do, we distribute the tissue across the United States. We have like over 50 collaborators. Like after the autopsy, usually we have another 6 to 8 hours to pack the tissue, process it, and send it around. And then usually the tissue gets there overnight. And and then there is a new team that spends another 8 hours, sorting cells and extracting RNA. It's an incredible resource and opportunity for so many people. So I always like to tell the story of at least some of our study participants, and these are two very special last gift participants who agreed to have their pictures and names shared. So on the left, that's Tony Bennett. I always like to do the joke, but I think it doesn't work here, that he always likes to be more famous of the other Tony Bennett, and Tony Bennett is a very famous country singer in the US. But he was last gift number 1, and he was extremely patient because at the time we didn't know exactly what we were doing, and there was a lot that we were figuring out. And here is him in a picture with his husband and another family member. And then the other study participant, his name was Michael Max Danielson. He was a publicist in Hollywood. He was the funniest guy ever, and I loved just going and hanging out with him on his patio and listening to all his stories about Angelina Jolie and Denzel Washington, and he knew everyone. And both participants were so involved in the last gift, and for them it was so important to be part of this study and to be able to give back this gift to their communities. So we have a very big social behavioral and ethics component to the last gift, and it's single-handed driven by Karine Dubé. She is a social behavioral scientist with a strong interest of ethics. And she wrote, this is not even all the paper that she wrote, she's very prolific, and about ethical consequences and lessons learned from the last gift. And I like to really think that the last gift pioneered end-of-life research in HIV in the United States. And now there are a few other sites that are replicating or trying to replicate the last gift. And we like to share, and we meet with people so that they don't have to basically start from scratch to set up, because we learned a lot over the last 7 years. Just to give you a few of the idea, this is one paper where Karine and her team did survey and focus group with the last gift participant, and really everyone without any exception was extremely happy to be part of the last gift. For them it was a legacy. It was about dignity, hope, and being able to give back to the community. In fact, we recently published a paper where we showed that people that are enrolled in the last gift, they experience an increase in quality of life at the end of life. Because I do think that the last gift people and team, because we really spend a lot of time with them, and for them it's so important to be able to really give back to the community. And these are some quotes from last gift participant where they really say, even if it doesn't cure HIV, I think it helps people from a mental standpoint, and that's positive. Another participant said, my death will not be in vain. It will allow me to leave a positive legacy, inspire others to give back. It's very powerful, and this is why I think I'm so proud to be part of Last Gift. And when we started it, we didn't really realize how big of an impact it might have. So Karine and her team also interviewed all of the Last Gift next of kin, and I do have to say also the next of kin share similar feelings, like they love seeing how being part of the last gift affects their loved one. For them, they really appreciate the last gift professionalism, the support that we provide. We really, I think we do a very good job in reaching out to the family, because we understand that really we rely on the family to call us at the time of that. So we try to create this relationship with the next of kin and the loved one, so that everyone is on the same page. And at the time of death, most of the time, that's the first thing that they do is to call us, because they know how important it is to start the process. And then we also had focus group and survey with the last gift clinical and rapid research autopsy staff. And I think everyone that is part of Last Gift, they will say the same. It's kind of really life-changing experience. It gives so much meaning to us. And even if it's not easy to wake up in the middle of the night, and I am not lying, we still feel that it's our responsibility to be there and fulfill their wishes. And I think I always like to say, a lot of my technicians, they say that now they don't even go out of the house or at the party without an extra pair of shoes and their scraps in the car, because everyone is so committed to Last Gift. We also, another little paper that I'm very proud of is that thanks to the Last Gift, I think that we also redefined the concept of vulnerability in the setting of end of life. Because when we started the Last Gift, there are some reviewers that were concerned that maybe we are, reaching out to people that are vulnerable at the end of life. And the community, and these are Jeff Taylor and Andy Kate, who are two of our community representatives that are part of the study team, they really pushed back and they say that's not true, right? Why, even if people are dying, as long as they are able to consent, we're not vulnerable. And it's also very patronizing to try to take away the opportunity for people to be part of research only because somebody's dying. And so the Last Gift was really designed from the very beginning as a participatory research model, because the community and the study participants were always part of every decision. And we wrote this little opinion piece, which I think everyone should read, where we really try to redefine the concept of vulnerability as an opportunity for communication, contemplation, and reflection, and not just an excuse to add a layer of, complication to the process. And really, if somebody wants to study a group that are potentially vulnerable, it's just an opportunity to reach out to the community leader and try to start a discussion about how these studies should be designed to make sure that everyone feels safe to be part of it. Okay, I wanted to now spend the last 10 to 12 minutes going through some of the findings of the Last Gift. And the Last Gift has been a lot of work. As you can imagine, it takes some time until the whole rapid research autopsy program was up and running, and we had to start enrolling people. And then we had COVID, so we couldn't really access people at the end of life because all of the hospice were shut down. And so recently we started publishing, and much more is coming. I think the last two CROI we had 8 abstracts accepted, but these are kind of the 4 papers that to me are right now the most meaningful from a biological standpoint. And the first one, so when we get the tissue, what do we do? So first of all, we do measure HIV DNA and cell-associated RNA in every tissue. And this is really the most granular study across tissues. And here you can see, for example, these are 2 participants. The first one on the top, you can see that he had very high HIV DNA in pancreas, and in fact, he had a pancreatic cancer. And the other participant on the bottom, he had more reservoir in the gut and in lymph nodes, which is more the expected. So I think that one thing that we learned from the Last Gift is that every story is unique, that every participant has kind of a unique distribution that is also the summary of their story, their lived experience, their diseases. And we also characterize the reservoir in the brain across every brain regions. And you can see in this figure where we found, that was a little bit of a surprise, that the HIV DNA was particularly high in the spinal cord. And we have one of our trainees has an oral at CROI for those of you that will go in March, and he will present how the reservoir in the spinal cord looks like, which is like a site that not many people have looked at. And now with the last gift, we have really the opportunity to dig into these reservoirs. Then what do we do next? We do sequence the virus, and when you sequence the virus, you're able to create this phylogenetic tree where we can look at the relation of the virus across participants within various compartments. And here you can see an example of a phylogenetic trees. And you can see like all of the color are intermixed. It looks like the virus, there are clones of virus that are across the compartment the same, which so we do have compartmentalized virus. It's not the same everywhere, but there are also shared virus across compartment, which suggests that maybe there is some migration of virus or cells with integrated virus across the body. And one of my colleagues, Dr. Shayon, he developed this phylogenetic and Bayesian modeling where he looks at migration of HIV across the body. And he does these very nice figures where he can show that even in people that are on antiretroviral therapy, there are some predicted migration across tissues and also between brain and the periphery and brain. And the hypothesis is that some immune cells that are infected with HIV, even when people are suppressed, they can still wander around the body. So this is evidence that the reservoir is not static, even in people that are on suppressive antiretroviral therapy, but there is still some movement and some migration across the body. Another application from HIV sequencing is that we can use those sequences to predict resistance to some of the neutralizing antibody or to predict how the HIV is adapting to the immune environment. And in this paper, it's again Antoine, he created a machine learning prediction model to investigate the distribution of susceptibility to viral neutralizing antibody, and he selected some antibody that are relevant to HIV cure studies, and he looked almost 800 viral sequences across 33 tissues. And what he saw is that the neutralization susceptibility of the individual neutralizing antibody were very heterogeneous. So within a person, there are resistant viruses, but overall, it was homogeneous across tissue. But there were some exceptions, and here you can see that in particular in the brain, we were able to find some provirus that was, and you can see it here, was predicted to be resistant to some of the neutralizing antibody, which means that if we use neutralizing antibody as part of curative strategies, and we only measure resistance pattern in blood, we might miss some virus that is resistant in the brain. And if we are really unlucky, it could then serve as a rebounding source. Another paper, which is probably my favorite so far, from Dr. Yang and Teng, these are our collaborators in North Carolina. And what we do, like when we do our rapid research autopsy, we send pieces of brain to them, and then they stay up for another 12 to 14 hours, and they sort immune cell from fresh brain. And they have been trying to do it for a very long time, and really was only after getting access to last gift that they finally were able to have brain the quality enough to be able to sort viable immune cells. And they sorted both microglia and T cells. And then what they do is they mix this immune cell with T cell from people without HIV, and they try to grow the virus. And they were able to grow virus from microglia, but not from T cells. And even when they analyzed the virus that grew, they were able to show that it was, a macrophage tropic virus. So there was no contamination from T cell. And so I think that this is probably the one most definitive evidence of the brain being a reservoir of replication-competent virus. And the same team has been studying, using the microglia from last gift in vitro and ex vivo to study mechanism of latency. They were able to show that the epigenetic mechanism of latency, they are different in microglia compared to T cell in the periphery. And they got too big a grant from the NIH right now to dig into these differences. Because again, like if we try to cure the virus, for example, with kick and kill strategies, it might be different in the brain and in the periphery. And this is another paper from Dr. Shaletsky, who is a collaborator of UCSD, where he sorted microglia and then he did single-cell RNA-seq and single-cell attack-seq, and he was able to capture some cells that express DNA and RNA and then analyze the immunologic profile. And he was able to show that microglia that expressed RNA have a different phenotype where they have more interferon-stimulated gene, which is also like a very unique finding and shows that cells that are infected have a different immune signature in the brain. We're also collaborating with a different team at Gladstone Institute, and they are running CyTOF, which is another very sophisticated technology where we can deeply characterize immune cells across the tissues, and they were able to demonstrate that there is different immune signature and different restriction factor in T cell across various tissue compartments. And this paper is also submitted and in revision right now in Cell Report. So in conclusion, I think that the last gift is really a unique opportunity to advance clinical and translational study in the setting on HIV cure. And but what also makes me really proud and what is important is that the last gift provides a framework to do end-of-life research in an ethical way, through this participatory research model where the study participant and the community are really empowered to be part of the study and every decision, and where we were able to challenge this paternalistic impulse of vulnerability, where a lot of reviewers were trying to shut down the last gift because they thought that it was not ethical to do. We also recognize that one cure might not fit all, that the last gift is, every participant is different, but really we only have a limited number of women or people of color, which is why we made a big effort to try to diversify our program. And we are now reaching out to international sites to try to enroll more women and people of color. Future direction, right? We have been talking with the community about what are the next steps for last gift. And there is a big push to try to do intervention at the end of life, because really we feel that the last gift is a unique opportunity to be able to do an intervention and then a rapid autopsy right away, and to be able to study the distribution of medication or compounds across the tissues and also across the blood-brain barrier. And we have done some foundational work with a survey and focus group, and it's incredible, like the vast majority of our participants will be willing to participate to end-of-life interventional study, which include a neutralizing antibody or even more, more investigational compounds. And we are right now really trying to work with the FDA and with some of our industry partners to try to find the right track to design such a study. But really, our community partners really like to say that these participants are dying to be able to make a difference. And so we should really try to give them every opportunity we can to participate to HIV cure research. So obviously the last gift, it's a big team, and here I just added some picture, but that's really only a small part of the group, which, there is technician and physician and outreach people and our study participant and the next of kin and community representative. And, it's kind of really my biggest study and the one that makes me the happiest every day when I go to work. And here, again, it's just a part of the team, and I feel like I always forget someone, but it's a big team, and everyone is very committed. Thank you.